Hello dear students, in this lecture we are going to see the point called as mapping by co-transduction. We have started our transduction chapter of genetic subject. In our last lecture we have discussed what is transduction, the mechanism of transduction, that is generalized and specialized transduction. In this lecture, we will discuss our last point of chapter, that is mapping by co-transduction. Now, what is mapping, or we can say what is gene mapping? It is defined as a method which is used to identify the location of a gene and the distance between the genes, how close the genes are present or how far the genes are present, what is the location of that gene, is all been studied under the definition of gene mapping. Okay, so before going to the co-transduction, let's understand the discovery behind the transduction. Okay, how the transduction came into existence. So we know that transduction was discovered by the scientist Joshua Ledberg and Norton Zinder in the year 1952. What they did, they carry out one experiment by using two strains of Asst.Prof.P.K.Gakkhad. Okay, they carry out the experiment by using the two strains of bacteria called Asst.Prof.P.K.Gakkhad. One strain was unable to produce phenylalanine, tryptophan, and was able to produce methionine and histidine amino acid. Okay, and the other strain B, was opposite of strain A. It was able to produce phenylalanine, tryptophan, and unable to produce methionine and histidine. Okay, so these two strains were there. So what he did, he took the two strains and put that two strains in the tube, and the shape of that tube was like a U. Okay, capital U, hence it is also called as U-tube experiment. So this is the U-shaped tube, and he placed one strain in one arm of the tube and other strain in another arm of the tube, and it was closed with the cotton plug. Pressure and suction was applied so that movement will be back and forth. Okay, and these two arms was separated by the filter. Okay, that is called as the filtrable agent, and that filter, it has the pore size of 0.2 micron, which was able to pass the genetic material from one arm to the another arm. Second, it was able to pass the virus. Third, it was able to pass the fluid, okay, liquid fluid, and it was unable to pass the bacteria. ये क्या ट्रांसफ़र कर सकता था, ये क्या पास कर सकता था? ये पास कर सकता था कोई भी जेनेटिक मटेरियल, कोई भी वायरस और लिक्विड कंटेंट. और ये क्या नहीं पास कर सकता था? इट वॉज अनेबल टू पास द बैक्टीरियल स्ट्रेन. ओके, सो what was happening in this U-tube? The strain which was able to produce phenylalanine, tryptophan, and unable to produce methionine and histidine was named as LA2 strain, and the strain which was unable to produce phenylalanine, tryptophan, and able to produce methionine and histidine was named as LA22. Okay, so the name was given as LA2 and LA22. So this negative, it indicates that it is a auxotropic strain. Okay, auxotropic strain means what? They lack something. Okay, or they lack something to produce. That is known as auxotropic. Means they are unable to grow in the nutrient media that is lacking in something. And opposite is the prototropic strain. Prototropic strain, they are able to grow in every situation. Okay, means they have their own gene to produce that amino acid, and auxotropic strain, they lack that gene. Means this La22 strain, they lack phenylalanine gene, they lack tryptophan gene, but they are able to produce this 2 amino acid. Okay, so that is the auxotropic. And prototropic means they have the gene to produce every amino acid. That is the difference between auxotropic and prototropic strain. Okay, so this was the preparation of U-tube, and here stopper is been fixed where pressure and suction is applied. When pressure is applied, there is movement of materials back and forth, आगे पीछे ये भागते रहते हैं. Okay, whatever the content has been present inside the tube. So what they did, after some time, when this experiment was the running was over, they took this or they grow into this onto this plate. Okay, they took this media and grow onto this Na plate. So what they noticed, in the first plate there was colony, and in the second plate there was no colony. मतलब यहाँ का जो उन्होंने कंटेंट लिया of the first tube, they saw that the colony was developed, but second arm tube there was no development of colony. Okay, so what they notice that there is something which is transferring the genetic material, and that transferring agent was your phage. Why? Because this filter has the ability to transfer the phage, okay, to transfer the virus, to transfer the genetic material, but it was unable to transfer the bacterial strain. Okay, so they noticed that there is something which can transfer the genetic material and can convert the auxotropic strain into prototropic strain. Okay, so in this plate the colonies were able to produce all the 4 amino acids called phenylalanine, tryptophan, methionine and histidine. As ये तो ऑलरेडी मिथियोनिन और हिस्टिडीन प्रोड्यूस कर सकते थे, लेकिन यहाँ का जो मटेरियल है वो फर्स्ट आर्म ट्यूब में ट्रांसफर हुआ है, हेंस दे आर ऑल्सो एबल टू प्रोड्यूस दिस phenylalanine and tryptophan amino acid. So this was the orientation of the U-tube experiment, or we can say this was the start point for the transduction, and then further different experiments were performed, and then it was finally concluded that that was the phage which was carrying something, which was carrying the genetic material from one strain to the another strain, and hence the definition comes like that. This transduction is defined as the transfer of genetic material with the help of the phage. Okay, so now what is co-transduction? Co means together. Okay, co means coordination, co means together, साथ. So co-transduction, it is simply defined as transfer of genetic material together. That is called as the co-transduction. So if 2 genes are close together, अगर 2 genes ये closely present है, a virus, a bacteriophage may pack a single piece of chromosome that carries both the genes and transfer that piece to the another bacterium. Okay, so if genetic material are placed in a close proximity, what that phage will do, that phage will carry both the genetic material to the another bacteria. Okay, so that is the co-transduction. Together वो pack करेगा और transfer करेगा from one cell to the another cell. So this co-transduction is possible only when genes are placed near or genes are placed at close proximity, but not possible when genes are placed very far away. Okay, so when genes are placed at a far away distance, there is no co-transduction, but if genes are present nearer, then the chances are there that co-transduction can take place. Okay, if I'll tell you the example of co-transduction, just now we have discussed the U-tube experiment, you can correlate that experiment with this co-transduction. So I have told you this first U-tube experiment. So Now in YouTube experiment, if we will carry out this experiment as an example of co-transduction, suppose we have seen that strain A was unable to produce phenylalanine tryptophan and able to produce methionine and histidine, and strain B was opposite. Okay, now suppose this two genes are placed at close proximity. Means what? Two strands are there. Okay, strand 1 is there and strand 1 is there and strand 2 is there. Okay, and strand 1 is unable to produce phenylalanine tryptophan and is able to produce methionine and histidine. Okay, this is your strain 1, and strain 2 is opposite. How it is? It is able to produce phenylalanine, able to produce tryptophan and unable to produce methionine and histidine. Okay, these are your two strains. And now here our bacteriophage is present. Okay, this is your bacteriophage. What it will do? It will transfer the gene which is able to produce this phenylalanine from the second strain. Okay, so it will take the phenylalanine strain and pass it to the first strain. What will happen? There is transduction. Okay, so what will happen? This first strain which was unable to produce phenylalanine will able to produce phenylalanine. Why? Because the genetic material from the second strain which was able to produce phenylalanine is transferred to the first strain which was unable to produce phenylalanine. Okay, hence it has been transduced, it will able to produce. Okay, and then it will become phenylalanine positive. So like that there is co-transduction. Okay, means what? Together the genetic material is transferred. Okay, so that is the concept of co-transduction. So I hope this might be clear to you. It is very simple. Transfer of genetic material together, that is called as the co-transduction. Thank you.